So what I want to do with the rest of our time today is talk about chapter 13, gene expression. I'm going to talk about the first part of chapter 14, which I was going to do as part of the makeup lecture during lab because we have enough time. And I think it's better if I just go through that during lab. Then you'll all just have it. Okay. So, but we've got a big enough chunk of time I want to get started on regulating gene expression. Okay. So in this chapter, we're going to look at how we regulate that process we just looked at, right? Transcription and translation is how we make a protein and send those proteins out into the body to do jobs, right? But we don't need every protein all the time. So we're going to have to produce only the proteins we need when we need them, and we're going to be constantly sort of turning on and turning off the production of those proteins. So we want to look at that balance. Okay, we're going to talk about the regulation of gene expression in general. We're going to look at it in prokaryotes, and then we're going to look at it in eukaryotes in different levels. So regulating gene expression epigenetically in eukaryotes, regulating at the transcription level, and regulating at the translational and post-translational level. Okay. Okay. So we already defined gene expression as the genetic information as it flows from genes to proteins. Right? We have all these genes housed in the nucleus, and we're going to open up and look at them one gene at a time and copy down those instructions to make the proteins. Right? That's the link between genotype and phenotype. Gene regulation is essentially turning genes on and turning genes off. Okay? The signal comes, right? The student eats the frosting covered donut, like in our video that we just watched, and the signal comes, I need to regulate my blood sugar. So the body's like, all right, insulin it is, and we turn on insulin production. Okay, blood sugar falls back down. We don't need any more insulin right now. We turn it off. Okay, there's all of these proteins that we're producing are going to be like got like a major switchboard going on, turning things on, turning things off when we need them. This is also a feedback loop, right? Because genes carry the instructions to make the proteins. The presence of the proteins and the concentration of the proteins then turns the genes on and off, right? As protein concentration rises and we have enough, we turn off that gene. As protein concentration falls or we get a signal that we need more, we turn the gene on. So these things are going to be regulating each other. Just like we looked at the products in negative feedback, being able to shut down the enzymes, we're going to be regulating turning genes on and off with how much product we're making and whether we have enough on hand. Okay. All right. So I think that it's perfectly okay to laugh at celebrities, like they're public figures. So this is George Clooney, and we're going to look at how gene expression varies in George's body as he ages. Okay, so when we think about gene expression, the genes that you need as a developing embryo, as an infant, as a toddler, as a young child, a teenager, and an adult are different. We go through this really distinct developmental phase. So as an organism goes through its lifetime and it completes its developmental pathway, it's going to need different genes. Right? So here young George is sitting at the top of the slide. He's doing a lot of running and playing and growing, right? So he's going to need all of the genes associated with growth turned on. His bones are going to be actively elongating. His organs are getting larger. His brain is making more connections and continuing to grow somewhat in size, right? All of those genes that are associated with growth and development are working really hard to help this little boy grow into a teenager, right? And then puberty hits, right? And for those of us, maybe some of us, it was longer ago than others, but all sorts of crazy stuff happens. There's a huge developmental switch when puberty happens. Genes that have never worked before are turned on. Hormones that you haven't used very much or have had in low background concentrations skyrocket, and crazy things happen to your body. Right? There's this developmental window where this whole other set of things are needed. And some of those you're going to continue to need as you move into adulthood. Others, once your secondary sex characteristics have developed and all of those things have been turned on, those genes will scale back a little bit, right? George isn't our young memory from ER anymore. Now he's quite a bit older. And so as you come to the end of your reproductive life, a lot of those hormones that you use during your reproductive years start to crank back down, right? Concentrations drop, and those genes are going to be downregulated. Okay, so as you go through this lifetime developmental process, the genes that you need at any moment in time are going to be turned on relative to what's happening in your development. Okay, that's one aspect of time. The other aspect of time is we talked about how genes interact with the environment, right? So George goes to the beach and gets a little too much sun, and his body has to deal with that environmental stimulus. I kind of think this is actually a spray-on tan because it's really orange. But at any rate, you're always, in addition to where you're at in this developmental phase, always also interacting with the environmental conditions. Okay, an environmental stimulus comes that says, let's make more melanin to respond to the sun, or the environmental stimulus comes that, like, I haven't had a good meal in a really long time and we're going to have to start burning our energy in a different way, and change happens, okay, based on what's going on in the environment. Like if you're entering starvation. Yeah. So when you get into a starvation phase, you switch over and you start burning muscle. But you start eating, in some cases, organ tissue even. Yeah. Okay. Gene expression also varies in space, right? Every single one of your cells, with the exception of eggs and sperm, right? So any cell in your body that is neither an egg cell nor a sperm cell has all of the same genetic information, right? All 46 chromosomes are present. They're carrying the exact same information. So theoretically, every cell in your body has all the information it needs to do all the tasks, right? But not every cell in your body does all of the tasks, right? Certain cells do certain tasks, and that's because of differentiation. Remember when we talked about stem cells being able to become any kind of cell when they differentiate? Once those cells have differentiated, muscle cells are going to be doing muscle jobs. They're going to be creating more muscle cells. Skin cells are doing skin jobs, and they create more skin cells, right? Our muscle cells don't create skin cells. Our skin cells don't create bone cells, and they don't do the jobs of those other cells, right? Part of that differentiation process is actually turning on the suite of genes you need and turning off the suite of genes you don't need, okay? Some of those cells can actually revert back to stem cells if given extreme conditions in a lab, right? We've been able to convert some skin cells back to stem cells. So that information is still there, but the differentiation process sort of packs away the genes you don't need, activates the genes you need, and then that cell's functioning in that space in the body. Does this all make sense for the most part? Okay. So we're going to look at a simple case of gene regulation, and I promise it's not going to feel simple, but it actually is, which is how gene expression is regulated in prokaryotes. So things like bacteria. We're going to focus specifically on E. coli. It's what's studied the most. So we're going to talk about gene regulation for a known suite of genes that occur in E. coli. Okay. One of our videos, not the one we just watched, but one of the previous videos actually zoomed in on the E. coli living in the intestine, right? And so we're going to go back to that example, and we're going to talk about some E. coli that are living in your intestine. And in the video we just watched, we had a sugar frosted covered donut, right? This is a Danish, I think, right? I'm not very good with my fancy breakfast foods. So this Danish has lots of things in it, right? Mostly a lot of sugar, but it's also got carbohydrates in other forms, the bready part of it. It has some fruit, which is mostly sugar. But then it has the fat, which is all the butter that was cut into the flour to make it all flaky and crisp, right? So in order to break down the sugars, we need sucrases, And to break down the starches, we need amylases. So those enzymes are going to go to work on the sugars and the starches. And some lipases are going to work on the fats, right? And we're going to break down this meal. And as your digestive system is breaking down this meal and moving nutrients down through the intestines and out through the intestinal walls to get to the bloodstream, the E. coli living in your gut are taking their share, right? They like sugar. They're going to be pulling those sugar molecules out of your bloodstream and eating them, right? In return, they give us a bunch of vitamins that we're not capable of making ourselves and do other things and help us break down some nutrients we can't break down. And so we have a symbiotic relationship with these E. coli that are living in our guts, right? E. coli is bad when it gets in the wrong place. So E. coli in the intestines, no problem. E. coli in the bladder, not so good. Yep. So it's all about where it's at. You can also get these super E. coli strains, which are these superbugs that come from factory farming. And that's what ends up when you have E. coli infested meats. There are these antibiotic resistant superbugs. They're not what normally lives healthily in your gut bacteria. Okay. I'm a Midwesterner, so if I were to eat that Danish, I would totally follow it with a glass of milk. I'd eat them, I would actually have them simultaneously because Midwesterners, we like our milk, whole milk, preferably with the cream on the top, right? So when you drink that glass of milk, you need a different set of enzymes to break down that milk. You need lactases to break down milk sugars, which are different than the sugars that were in our fruit and the sugars that were in our granulated sugar that was part of the Danish, right? E. coli is like, I don't really need, I don't like lactase or lactose that much. I'd rather not eat milk. But if that's what you're going to feed me, because I live in a Midwesterner's gut, then I'm going to have to turn on some special machinery in order to break down those lactose molecules and use that as a food source as well. So generally speaking, our E. coli really wants us to just eat sugar. But if we give it milk to digest instead, then the bacteria's system will respond. It'll turn on the genes it needs to break down lactose so that it can utilize that food source. Okay? And the E. coli that lives in your intestine is constantly having to shift what it's doing based on what you eat, right? Because its only way to get food is what comes through your intestine. And so if you go on a sugar binge, it's going to be super happy. It's just lapping up the sugar. But if you have a midnight milk snack, it's like, oh, I'm kind of hungry. I guess I'm going to have to eat that too. And so it's going to have to adjust, all right? So that's the story for this series of lactose utilization genes. So this picture is something that we're going to look at for the next 10 minutes or so. So I'm going to walk you through what is displayed here, and then we will add to it as we go through the story. So in blue, out here on the end, right, this is our E. coli's DNA. And all of this is DNA, okay? We've just blocked off some sections that have particular functions so that we can talk about them independently. So here in dark blue, we have what's called a regulatory gene. In green, we have something that's called the promoter. Here we have something called the operator. And then here we have 1, 2, 3 lactose utilization genes. And those 3 genes, all 3 of them are necessary for E. coli to gain nutrition from lactose. Okay. The rest of these pictures are separate from the DNA. Here we have the mRNA. And so this is the message that is made by the regulatory gene. And the message made by the regulatory gene makes something called a repressor. And so this repressor protein can go up and interact with the DNA. Okay. Clean that up. The regulatory gene is outside of what we call the operon. And the operon is all of this. The promoter, the operator, and the 3 lactose utilization genes. In these prokaryotes, it's really convenient. We need these 3 genes all at the same time to break down lactose. They're right next to each other in the organism's genome, one right after the other, and they're actually transcribed and translated as a set. Okay, so they're all packaged up so that when lactose comes in, we say turn on the lactose operon, and we can digest that food source. Okay, so the 3 lactose utilization genes are referred to as structural genes, as opposed to the regulatory gene, which makes a regulatory protein. Okay, that's not ever going to test you on that, but that's what's there. These 3 lactose utilization genes produce 3 separate enzymes. So when we individually transcribe and translate them, we're going to make 3 separate enzymes, and those 3 enzymes do different jobs. One has to do with how we move lactose around, one has to do with creating the enzyme that breaks down lactose, and another one has to do with moving the products through the organism's cell. Anyway, we need all 3 in order to metabolize lactose effectively. The promoter is the place where the RNA polymerase binds. We've used the term promoter before when we talked about transcription. It's where we initiate transcription. So the RNA polymerase has to come and bind to the promoter in order for transcription to get started. Okay. When the RNA polymerase binds to the promoter, all 3 lactose utilization genes will be transcribed as a set. Okay. The operator is essentially the on-off switch. And so the operator is going to be able to block transcription of the lactose utilization genes. So if our repressor protein from our regulatory gene is attached to the operator, and RNA polymerase tries to come up and bind to the promoter, this repressor protein is actually blocking the promoter. So RNA polymerase is either unable to bind, or if it's able to bind, as it starts to move forward, it gets knocked right off. Okay, so the operator is going to function like an on-off switch. When the operator is blocked, transcription is off. When the operator is unblocked or open, transcription is on. Okay. Operons are only found in prokaryotes. When we talk about eukaryotic gene regulation, we might have a suite of genes that need to be transcribed at the same time, and they're found on a dozen different chromosomes, right? They're not necessarily packaged together in a suite like this. We only see this in prokaryotes. This is not the only operon that's been described. There are many operons are known, and we understand how they function. I'm only going through one example. I think actually you might have 2 examples in your book. I'm just going through one of them. Okay. The operon contains the promoter, the operator, and the structural genes. It allows for a single on-off switch for the transcription of all the structural genes. So it makes it convenient to utilize these genes as a package. Okay. This particular operon is known as the lac operon, and lac comes from lactose because it has to deal with utilizing lactose as a nutrition source. Normally, because E. coli would really rather not eat lactose, they want to just gulp down sugar if they can, their lac operon is going to be by default in the off position. It's going to be blocked. They're not making any lactose utilization genes until they haven't had a meal in a while, and you drink a glass of milk and they're like, okay, I'm hungry, I guess I'm going to have to eat this milk, and they turn on the lactose utilization genes. Okay. So our normal default state is for this repressor protein to be blocking the operator, or attached to the operator, and blocking the promoter so RNA polymerase can't bind. That's the default state in E. coli in our intestines. Okay. The repressor protein that is made by this regulatory gene, the regulatory gene is what we call upstream before the structural genes. It's completely separate and it's outside of the operon. So we don't have to interact or do anything at the operon for the repressor protein to be transcribed, translated, right? It's going to be happening completely separate. it's going to typically, whenever it's created, bind to the operator and turn off the lac operon, right? Its creation is controlled by this regulatory gene, and generally speaking, the repressor proteins are just made whenever they're in short supply. So when we run out of repressor proteins, we make some more, and we would run out because they get old and get broken down. Okay. These are our two positions for the regulatory protein. Typically, it's active. In its active form, it has this shape, and it adheres to the operator. Okay. You'll notice, I haven't drawn your attention to it yet, but there's a groove in this protein. Right? Remember, proteins are three-dimensional shapes. Just like our enzymes, they have spaces where other things can come in and interact. And that groove is exactly the right size and shape for a molecule of lactose. So when all of a sudden the E. coli's body is flooded with lactose that it's bringing in, all of those lactose molecules go and bind up the repressors. And when the lactose binds to the repressor, it changes its shape. It loses its little fingers that help it to attach to the operator, and it falls off of the operator. So as lactose concentration increases, our repressor proteins are inactivated, and now the operator is empty, and our RNA polymerase can bind to the promoter and transcribe. Okay. So when the repressor protein is inactive, RNA polymerase binds, and it transcribes all three structural genes. They are transcribed as one, but they have multiple stop and start codons, so they're translated into three separate proteins that do three separate jobs. Okay. But it's all one transcription. So we just turn this on once, transcribe the whole thing, and then we're going to translate it into the three genes that we need, or the three proteins that we need. Okay. What do you think happens when these genes are transcribed inside the E. coli's body? What's it going to do with those genes? Well, yep, it makes protein. What's it going to do with those proteins? Good job. I was using the wrong word, huh? Break down the lactose. Right? The E. coli is going to go eat a bunch of lactose. As it's eating up the lactose, what happens to our lactose is in our repressor proteins. They fall out and get eaten. So then our repressor protein goes back to its original shape, and it turns off the genes, just like a switch. So lactose is metabolized, lactose concentration falls, the repressor proteins reactivate, and transcription stops, because now we don't need these genes anymore. Okay. They probably have a short lifespan and linger around for a little while, and then if we're not using the proteins, they'll get broken down and recycled. Okay. Until the next class of milk comes along. All right. Does that make sense? Yeah? Mm-hmm. It doesn't actually have anything to do with E. coli in the gut. If you're lactose intolerant, you don't make the right enzymes to break down lactose. And so part of the like Lactaid, right, is to try to give you the right enzymes to help you break it down. Okay. Yeah, I don't know. And I don't know that much about the underlying mechanism of lactose intolerance. But it might be an example in this textbook. If it's not, it's an example in another textbook. We could look it up pretty easily because it's known. It's just not something I know off the top of my head. Okay, so I want to move on to talking a little bit about gene regulation in eukaryotes. I feel like I should do this material at the beginning of the semester. You guys just too worn out or something. It's a little dense. You guys are like, can I just take a nap? Huh? At what point? After the second exam? You were so into it in the beginning.
I'm still into it.
Okay. So the next section we're going to talk about is about something called epigenetics. And how many of you have heard this term epigenetics before? Where did you hear it? Huh? Here? Epigenetics. In a class? Okay. I feel like we hear it on, like, if you listen to the news, especially like if you're somebody who like listens to NPR while you're commuting, I feel like you're likely to hear a story about epigenetics every couple weeks at least, just by accident. Epigenetics is a really hot area of research right now. It's something that we didn't know happened until recently-ish, say the last decade or so. And so it's an area that, like, once we started to understand it, became something people really wanted to research a lot. So there's a lot of work going on in epigenetics now, trying to understand exactly how it happens. So what I'm going to tell you now will probably be out of date within a couple of years, right? It's just an area of research that is moving really fast. So this is like where we're at now-ish, and unless something big happened last week that I missed, and know that epigenetics is something we're continuing to understand better and better because it's a very active area of research. Okay. Do you have a question? Okay. So a couple of chapters ago, we talked about cell differentiation, and I just referred to it a few minutes ago, right? This is where our little lump of stem cells starts to take on the characteristics of cell lines that are going to do specific tasks. Some cells become nerve cells, blood cells, muscle cells. They take on a particular structure and function, right? That's established. In order to maintain that cell differentiation, some genes have to be expressed and other genes are going to not be expressed. Some genes are going to be turned on, other genes are going to be turned off, right? I just sort of referenced all of this in our George Clooney example. That unique gene expression is going to be maintained over time throughout the organism's lifespan until some signal comes in and disrupts that, right? Maybe that's a developmental signal. Oh, hey, puberty's here, time to change things up a little bit. Or, oh, hey, menopause is here, change again, right? Whatever it is that comes in and causes a change. Or there's an environmental change, right? We already kind of referred to all of this. I'm just laying it out. One of the ways that we can regulate what genes are actively being used or not being used in a cell is through nucleosomes. And you've heard this term before. The nucleosome is the little bead of DNA that's created when we wind DNA around a histone block, right? Remember I talked about it being like thread on a bobbin? We're going to wind up some of that DNA, and in doing so, we could be preparing for mitosis, preparing for travel, right? We're going to move and we need to package up the DNA, or we could just be getting it out of the way. Okay? So I think of packaging genes up into nucleosomes as being kind of like putting things in the attic, right? They're still there. You can access them if you need to, but they're not out in your everyday life, right? So sections of the genome that aren't being used at this moment in time are going to be wrapped around some histone proteins into a little nucleosome and put in the attic, set aside, right? But the nucleosomes can move, so they can slide back and forth on the DNA, so we can open up those boxes and get out the information if we need it, okay? They just hang out on our DNA, help keeping it organized, because it's just a giant spaghetti pile, right? Our DNA is so much bigger than our actual cell if you stretched it all out. We got to fit it in there somehow. Okay. Yeah. Next thing that I want to talk about to complete this story is the process of methylation. And when we were talking about DNA replication, I said some of the editing mechanisms know which is the new strand until that strand has been completed, right? And one of the things about the completion process is actually replicating the methylation pattern. And the methylation is just adding a methyl group. Does anybody remember what a methyl group is? What's a methyl group? Carbon and hydrogen, right? So here, this cytosine does not have a methyl group on it. Here, 5-methylcytosine has a methyl group that has been added to it. Okay, so we can add those methyl groups to our DNA, to our chromatin in the cell, and what do you think that might do? It creates an obstruction. When we add that methyl group, it changes the shape of our individual nucleotide just a little bit. It can functionally block transcription so that when the transcription enzymes get there, they bump into that methyl group and they fall off, or they can lose their affinity to bind because of the methyl group. And also, if we have our nucleosomes close together, they can't slide as easily, and so they get blocked up, and we can kind of hold them in place by adding methyl groups to them to keep them inaccessible and to inactivate a gene long term. So we really want to, this cell is going to be a skin cell. We don't need the genes to make muscle fiber. We're just going to package them up, pack all the nucleosomes close together, methylate them, and now they're just in the attic and forgotten about. We're not going to access them. Okay. Methyl groups are common. I don't know, like they're common. That could be why that they're used. I don't know. I'm just guessing. Do you have a question? No, just thinking. Okay. So when those nucleosomes are close together, and then we've added those methyl groups to them, those genes are inactive. They're not going to be accessible. And the only way to access them is to unpackage all of that stuff, right? And get back in there. That can happen if the right signals come. But for now, they're set aside. Okay. So when we use this term epigenetics, and we talk about patterns of inheritance that are epigenetic, we're talking about times when there is a change in the gene expression without a change in the nucleotide sequence. So using methyl groups, using nucleosomes in DNA packaging, causes us to change how the genes are expressed, put some of them away, turn others on, right? But it doesn't require a mutation for that gene expression to change. And these things that we just talked about can also be undone. They can be reversed. So if you have a change in gene expression due to a mutation, that's permanent. We can't unmutate our cell, right? But if we have an epigenetic pattern that's applied, that can be reversed if developmental or environmental signals come along later that cause a change. The cool thing about epigenetic patterns is they can be inherited. And so one of the first studies that started to show this looked back at medical and birth and death records in a community during a time when their health and survival was very much linked to food availability. So before we had such a globalized food economy, and if we had a good year, people got fat and they lived. If we had a bad year, some kids died of starvation, right? And so if you were born during a fat year and your parents were packing it on and food was plentiful, your risk of all sorts of obesity-related health disorders is higher than if you were born during a lean year when your parents were dealing with a starvation situation. And in further studies, they started to unpack that some of that comes from these epigenetic patterns. And so the condition that an individual was in can be passed on to their offspring, and the offspring is prepared then to experience the same condition. And it can actually even go to the next generation. Unless a strong enough signal comes in to reverse it, we're all just preparing for the condition our grandparents and great-grandparents found themselves in that was extreme enough to change your DNA patterning. Yeah, survivability. So these epigenetic patterns can be inherited for multiple generations unless a signal comes along to reverse them. So this is definitely an area of a lot of research that's going on. And when we talk about evolution, we're really always talking about natural selection and Darwinian evolution. And this is actually like something that can change in your lifetime. You can acquire a new epigenetic pattern and then pass it on. That's really kind of unique and not how we thought evolution worked. So this is just sort of summarizing some of the things we know now. Epigenetic changes to the chromatin can happen because of things that you experience during development, like in utero or during childhood. Environmental chemicals, drugs, or pharmaceuticals that you're exposed to, signals that occur as a normal part of aging, and things that can happen because of what you eat in your diet and what your dietary patterns are. And it might result in some unpleasant things like cancer and autoimmune diseases and mental disorders and diabetes that are linked to some combinations of those epigenetic patterns that are a result of these sort of more environmental or experiential factors as opposed to raw genetic mutation or genetic factors, or well, they're genetic factors, but those, you know, sort of our typical sense of inheritance is expanding as we think about how these gene expressions can be affected by experiences in life. Okay. All right. Let's stop there. So we will finish chapter 13 on Monday.